very much. Such an honor, pleasure to be here and jump right in and talk about the origins of blood cancer. So we've done a huge amount as a community of science, community studying cancer, a huge amount of sequencing in the last 10 or 15 years and identifying most of the major driver mutations that drive cancer. And one of the major conclusions is that the vast, vast majority of human malignancies are caused by the sequential acquisition of multiple mutations leading to a full-blown malignancy. And under almost no circumstance does a single genetic lesion all by itself lead to full malignant transformation. And there's a couple conclusions you can draw from that, one of which is that if there are many steps on the way to getting a malignancy, say this is the blood system and each of these columns is the set of hematopoietic stem cells you have, there may be initial mutations that occur that lead to a clonal expansion that are not malignant. And then within that, one of the cells may get a second mutation leading to another clonal expansion. One of those cells will lead to another mutation leading to a further clonal expansion. So there's a few conclusions you can draw from that. One is that those initial clonal expansions with initial driver mutations may be orders of magnitude more common than full-blown malignancies. And another conclusion is that once a patient presents with a full-blown malignancy, it's a very genetically complicated bone marrow where there are cells with 5 mutations, cells with 4, 3, 2, 1. And when we treat a patient and they go into remission, that can mean many different things. It could mean that that final clone is gone, but all those precursor clones are still there. It could mean that all the clones are gone, and that has profound implications for how relapse occurs. So we decided that we wanted to understand that very first mutation that occurs, very similar to a polyp in a colon, but in the blood system where the cells circulate. And an advantage for the blood system for studying this broadly is that exome sequencing or genome sequencing or SNP arrays that are done to identify germline predisposition to various diseases is always done in the blood almost universally because it's the most easily accessible DNA from individuals. So when we first dove into this, the Broad Institute was just engaging in really large-scale exome sequencing projects and had sequenced at that time about 17,000 exomes from many different collaborations, many different projects. And we were able to aggregate those and to look for driver mutations, known genes that are recurrently mutated in blood cancers, and see, can we see those mutations in normal individuals, unselected for any hematologic phenotype, in these exome sequencing data. And what we found was that young individuals under the age of 40 almost never had any evidence of one of these mutations. But by the age of 70, on the order of 10% of everybody in the general population had a mutation in their exome sequencing data that was quite large, accounting for about 20% of all the circulating blood cells. So this is not a tiny, tiny clone. This is an enormous clone comprising a large percentage of the organ. And the mutations we found are all mutations that we see in acute myeloid leukemia as well as other myeloid malignancies, which is fairly surprising. AML is a lethal, aggressive disease, but these are mutations that are clearly not causing leukemia in everybody and are present in a large fraction of the population. So one question is this crazy or incredibly unusual that we would have all these clonal expansions? And one way to think about it, particularly to people who like screens like Hannes, is that we are constantly doing genome-wide selection screens in our bone marrow. And these mutations are effectively genome-wide screens for mutations that lead to clonal expansion over 70 years of human life. And the mutations end up saturating our genome much more than you might think. So from elegant work from Tim Lay's lab at WashU, where he sequenced individual hematopoietic stem cells from people of different age, we gain about 1 exonic mutation per hematopoietic stem cell per decade. So every one of our hematopoietic stem cells is mutating constantly, and some of those mutations will happen in the exome in a coding sequence, and we're constantly acquiring them. And the number of hematopoietic stem cells has been something people have debated for decades, but really elegant work from Peter Campbell at the Sanger Center, where they did genome sequencing of many, many hematopoietic stem cells, looked at the passenger mutations, and could calculate the number of hematopoietic stem cells we have with greater accuracy than previous, would indicate we have about 50 to 200,000 true hematopoietic stem cells in our bone marrow. And so that would imply, based on that math, that we get 25 to 100 mutations in every single gene during our lifetime by the age of 70. And so we are introducing mutations into every tumor suppressor and every oncogene in our genome. And then they are competing with each other. And then sometimes one will grow out and gain clonal dominance. So within each of our bone marrows, that competition is ongoing. And between individuals now, if we look at a population basis, we've now looked at well over 100,000 exomes from unselected individuals. We get a very, very complete picture of all of the somatic mutations that can lead to clonal dominance. The genes that lead that are largely epigenetic regulators. DNMT3A, TET2, ASXL1 are genes that regulate DNA methylation and epigenetics of hematopoietic stem cells. Exactly why they lead to that clonal dominance, we understand a lot about the particular changes in epigenetics or gene expression that occur with these mutations, but I don't think we really understand why these mutations lead to clonal dominance. P53 is actually relatively common. We think of P53 as this horrible gene that leads to the worst outcome in myelodysplastic syndrome or acute myeloid leukemia, but huge number of individuals in the world are walking around with P53 mutations in their bone marrow without a evident phenotype. And then JAK2, SF3B1, and others. Now, we know from, as I mentioned at the beginning, large-scale sequencing studies, almost all cancers, including MDS and leukemia, have multiple mutations. So in a standard leukemia sequencing study, there might be an average of 3 to 6 or 7 mutations per individual, per leukemia sample among the driver genes that we know about. In this data set, it's very different. Almost everybody had just a single mutation. So that's consistent again with this is just a pre-malignant state. This isn't actually MDS or AML that's just percolating. This is a distinct state. And consistent as well with this being a pre-malignant state, individuals who have a mutation in their blood, clonal hematopoiesis, have a much higher risk of getting a blood cancer, about a 10-fold increased risk if you take everybody. That risk has now been refined a little bit. If you have multiple mutations, if you have larger clones, if you have particular mutations, including P53, the risk is much higher. So the risk can be as high as 50 to 100-fold greater than the general population with, in particular scenarios with larger clones, et cetera. So it's a very good predictor of who might get a blood cancer. But unlike a polyp in the colon, which we can just clip out, and the risk of colon cancer completely goes away. There's obviously no surgical technique to remove 20% of the blood cells with one of these mutations. We don't even have a surface marker that we could sort some of them out. So we're going to need a pharmacologic intervention to decrease the clone size, which we don't have yet. So there's certainly no indication to screening the general population for cancer risk, for blood cancer, because we don't have an intervention, but we could at least identify those at high risk. One of the really fun aspects of this field on clonal hematopoiesis has been the very rapid convergence of many, many groups with almost identical findings. This is work from Steve McCarroll at Harvard Medical School, who was actually studying the genetics of schizophrenia, but ended up sequencing a large number of blood samples. He was interested in somatic mosaicism, ended up finding really the identical things. The same genes are mutated, same age association, the same increased risk of blood cancers. Other groups at the Sanger Center, at WashU, and many other places, and now all over the world have extremely similar findings. So this rapidly became a very robust finding. With all those studies, one of the things that came out is, perhaps not surprisingly, the deeper you sequence, the more of this you find. So if you do error-corrected, barcoded sequencing, so you can find 1 in 10,000 cells that bear a mutation, essentially all of us will have mutations in DNMT3A or TET2 as we get older. If you use whole genome sequencing at lower coverage, you find less of it. So not too surprising. But it starts to raise the question, given we find clinical consequences of these mutations, we need to somehow start to define the state. We took a first stab at that, calling it clonal hematopoiesis of indeterminate potential, or CHIP. The first criteria is that these individuals don't have a hematologic malignancy. And this, at the time, was actually quite an important distinction because pathologists would see a patient, maybe they'd have a little bit of anemia, and they'd find a DNMT3A mutation. They say, well, they have a mutation, they have anemia, they must have myelodysplastic syndrome, which is a disease with a poor survival, a relatively aggressive course. That is absolutely not the case. These DNMT3A mutations are happening in healthy individuals, and they are far from having a blood cancer. So without morphologic diagnostic evidence of a malignancy, they don't have one. They have a pre-malignant state. And we needed a threshold of how big the clone should be before we called it an entity. And we provisionally said it needed a variant allele fraction of at least 2%. That's open to revision. We may make it even a bigger clone is where we get nervous about it. But again, if we don't have that, essentially everybody will have this state, and it sort of loses its meaning. And with this definition, the odds of progression from the pre-malignant state to a full-blown malignancy is about 0.5 to 1% per year, which is very similar to other pre-malignant states like MGUS, which is the monoclonal gammopathy of undetermined significance, which is the pre-malignant state for multiple myeloma. So CHIP is this pre-malignant entity. Now, one of the types of data that we had for really large numbers of individuals was overall survival data. So that's one of the endpoints that you can get at a population scale in the US through the Social Security database or through other mechanisms. And we found that individuals with CHIP, with one of these mutant clones in their blood, had a markedly increased overall mortality, so increased risk of dying of all causes. And only one person in this cohort for which we had mortality data actually got a blood cancer. So this was shifting overall mortality quite significantly. A 40% increased risk of dying of all causes is a pretty big shift. Not that many individual diseases can actually shift by that much. Blood cancers, though we love to study them in the lab and develop new therapies, are relatively rare and quite difficult to shift overall mortality just on the basis of blood cancers. So we needed to think of what else could be driving this shift in mortality. One, and this has been a robust finding across many labs and many studies, many data sets. So one logical thing would be they're dying of cancer. We're seeing a mutation in their blood. Maybe they've got a different process of mutagenesis and they're more likely to get other mutations. And that turned out not to be the case. The mortality from solid tumors wasn't significantly different in this data set. But much to our surprise, these individuals with a mutation in their blood with CHIP had an increased risk of developing cardiovascular disease with an increased risk of both heart attacks and ischemic strokes. And the risk was pretty big. A hazard ratio of 2 is fairly large in this field. The hazard ratio in this data set or others for hypertension or high cholesterol is about 1.6, 1.8. So we think of having high blood pressure as a major risk factor for cardiovascular disease, and this was consistently showing an even greater risk. So this is a potentially substantial risk factor for cardiovascular disease. This certainly was not remotely on our radar. It wasn't an intentional analysis that we set out to do at the beginning of the study. So there's always statistical concerns about exploring around data sets with multiple hypotheses before you find something like this. So we spent the next few years validating this in 4 additional cohorts, and now it's been validated in many more, including a paper just came online this week in the UK Biobank and replicated this finding almost identically. So these are prospective cohorts, meaning blood is drawn at the beginning of the study. Patients were followed for years. Some patients got a heart attack. Some individuals didn't. We could compare the prevalence of CHIP in those who did or did not get heart attacks, and having CHIP at the beginning of the study about doubled the risk of a subsequent heart attack, an incident event. And that was statistically significant in all of our cohorts. We also looked at early onset MI. That is our people who had a heart attack in their 40s. And even though CHIP is really rare in those individuals, if you have CHIP in your 40s, it's more than 4-fold increased risk of subsequently developing a heart attack. So in general, you can't say that genetic association, however robust, is a causal association. And there was a lot of skepticism in our lab and elsewhere that this was fully causal. You could argue that maybe this is a great marker of aging, and we just picked up a better marker of aging in cardiovascular disease as a defining disease of aging, and that's what's accounting for it. Or there's some other variable that we haven't accounted for. The reason to think that it's not crazy that it could be a causal effect is that atherosclerosis that leads to heart attacks is driven by blood cells. It's driven by macrophages and monocytes and neutrophils and platelets, all of which will bear these mutations. So if you have a mutation in a hematopoietic stem cell, all of the progeny are going to have those mutations. And if you mutate a key epigenetic regulator, it's going to change the function of those cells. It'll change the gene expression in those cells. It'll change the response of those cells to stimuli. And so it's conceivable that these abnormal mutated blood cells are actually altering the development of atherosclerosis. So we wanted to ask that as an experimental question in the lab. It turns out that mice live very well. They don't get atherosclerosis. They eat healthy diets, so they won't get any evident atherosclerosis during the life expectancy of a postdoctoral fellow in the lab. So we used a model that is really well established in this field, the LDL receptor knockout mouse. So they get very high cholesterol. We feed them a Western diet, very high cholesterol, and they get high cholesterol levels. They get atherosclerosis in a matter of weeks to months. And we asked the question, if you only change the genotype of the blood, will you alter the development of atherosclerosis? So if you transplant in wild type hematopoietic cells, bone marrow cells, or TET2 mutant or DNMT3A mutant, ASXL1 mutant blood cells, do you alter the development of atherosclerosis? So we did that, and very robustly it did. So these are mice with wild type blood cells, and these are the atherosclerotic plaques, an oil red O stain that stains the lipids in the macrophages. This is with TET2 heterozygous knockout or TET2 homozygous knockout in the blood cells. This is the descending aorta, which is one of the areas that develops atherosclerosis in a mouse. So simply changing the genotype of the blood is enough to accelerate atherosclerosis at multiple time points. It turns out with multiple genotypes, and again now in multiple labs around the world. So at least in this mouse model, these mutant blood cells are sufficient to alter atherogenesis. So how could that be happening? We could narrow down using different Cree lines that this is a myeloid effect. It wasn't a platelet-driven effect. And we focused on macrophages, which was really the dominant cell type in these plaques. And we did some RNA sequencing on these macrophages. And these are actually TET2 versus wild type macrophages exposed to LDL cholesterol. And a number of genes are differentially expressed, but amongst them are some inflammatory cytokines here in red. And many of those cytokines had already been implicated in the biology of atherosclerosis. And perhaps one of the most interesting ones that was differentially expressed was interleukin-1 beta. We went on to show that interleukin-1 beta levels are elevated in the serum of mice with TET2 mutations, and we've now seen that IL-1 beta is elevated in the serum of humans with TET2 chip in their blood. So it's a robustly elevated gene. IL-1 beta is one of the cytokines that's been very well implicated in the biology of atherosclerosis, so much that an antibody was developed called canakinumab by Novartis. And they actually did a large clinical trial called the CANTO study with 10,000 individuals randomized to this drug blocking IL-1 beta or a placebo control, and it actually worked. It decreased cardiovascular endpoints. It met its endpoint, and that was published in the New England Journal almost at the same time as our paper. So we have a causal mechanism why interleukin-1 beta might be elevated in some individuals. They have shown that interleukin-1 beta antagonism can benefit patients with atherosclerosis. And when they went back, based on our findings, and sequenced a few thousand patients in their clinical trial, it looked like individuals with CHIP, particularly DNMT3A and TET2 mutations, had a major benefit from canakinumab, whereas those without a mutation did not have a significant benefit. So the next stage in this field is really to validate that, but clinically, whether this is a good biomarker for patients who might benefit from anti-IL-1 beta therapy. So that's the state of this field now, that these mutations occur in a hematopoietic stem cell. Those become a larger expanded clone. The progeny of those cells, those mutant cells, will circulate in the blood, so we'll have mutant neutrophils and monocytes and macrophages and platelets. Amongst other effects, they will elaborate higher levels of pro-inflammatory cytokines that will support atherosclerosis. Some of these cells will get into other organs, and that's a major area of research now and a variety of organs, but it's been shown now in the setting of heart failure as well as patients with aortic stenosis getting valve replacements, that CHIP is a major predictor of outcome in all of those settings. And so there's hopefully a lot more biology to understand about how these mutant cells are influencing human disease. And since these pro-inflammatory cytokines have been implicated in a variety of diseases of aging, the other major field now is to understand what other diseases associated with aging, other inflammatory diseases, rheumatologic diseases might also be influenced by CHIP. One of the other clinical scenarios that we think about a lot is therapy-related myeloid malignancies. So these are patients who get a solid tumor, get chemotherapy or radiation, and do great, are in full remission, but then go on to get a therapy-related MDS or AML. So it seems like the worst possible luck. They had breast cancer or other disease, did great, but then related to having received the therapy for their solid tumor, go on to get a horrible leukemia. These therapy-related leukemias tend to be refractory to therapy. They tend to be particularly fatal malignancies. But there's been a gradual change in the field of understanding therapy-related malignancies, that it may not be simply the mutagenic effect of the therapy that leads to these leukemias. It may be the selective pressure of these therapies for particular mutations. So we asked, in a patient population, we chose lymphoma initially, does the presence of a mutation at the time of therapy predict later therapy-related malignancies? So these are actually lymphoma patients who are getting autologous stem cell transplants. We get them very high-dose chemotherapy, and we bank some of their bone marrow cells, give them high-dose chemotherapy, and then we rescue their bone marrow with their own cells. And those patients have a very high rate of therapy-related malignancies, and it's a common procedure. So we did about 400 autologous transplants for lymphoma patients, non-Hodgkin's lymphoma patients, over 7 or 8 years just at our institution. So we asked whether having CHIP at the time of that autologous stem cell transplant predicted a subsequent therapy-related malignancy, and it did, so that it really dramatically increased the risk. Those without CHIP had a much lower rate of getting a therapy-related malignancies than those with CHIP, and that translated to a much worse overall survival. So that's one of the biggest now predictors of overall survival in individuals who are getting an autologous stem cell transplant for their lymphoma. Some of that mortality was due to the development of therapy-related malignancies that couldn't be cured, but actually some of that mortality was also due to atherosclerotic cardiovascular disease as well. So what are the genes that drive that process? P53 is one of the major ones. So a mutant P53 hematopoietic stem cell, when exposed to chemotherapy, will have a survival advantage compared to the wild-type cells. So that clone will grow out. That is super reproducible in mouse models or in vitro. But another gene turns out to be extremely commonly mutated. That's a gene called PPM1D. PPM1D is actually a phosphatase that regulates P53 as well as all the other phosphorylated proteins in the DNA damage response pathway. So this again is effectively a functional genomic screen of mutations that lead to outgrowth in the setting of chemotherapy, and P53 and PPM1D are the winners. We'd actually done another large-scale study of myelodysplastic syndrome patients, the genetics of MDS patients, and we specifically looked amongst the 1,500 MDS patients that we genetically characterized, what are the genes that are more commonly mutated in those who have therapy-related MDS compared to de novo MDS, and those were, again, the two genes that came out, PPM1D and P53. So these are the really dominant causes. So if we think about selective pressure, why some clones grow out, that selective pressure is dependent on the environment, and an old bone marrow might be different from an infant bone marrow, but the selective pressure of chemotherapy also dramatically changes clonal advantage, and P53 and PPM1D mutations are the dominant mutations that come out. Within the P53 world, we've done a fair bit of research recently on understanding the particular mutations and their cause, and showing that the missense mutations, the hotspot mutations in the DNA binding domain, which are so common, act as dominant negatives, and we can actually see a spectrum of the impact of p53 mutations and their selective pressure in CHIP after exposure to chemotherapy or radiation, where heterozygous loss causes some resistance to DNA damage, but a missense mutation causes even more resistance to DNA damage, and then either missense crossed to a null or a complete p53 null have equivalent resistance to DNA damage, and are have the least p53 activity and the most likelihood of progressing. I mentioned this other gene, ppm1d. ppm1d is this phosphatase. When we did phosphoproteomics, you see alteration in phosphosites throughout the DNA damage response pathway. We think about it as once the DNA damage response pathway is triggered, this whole pathway gets phosphorylated, and ppm1d is actually turned on. It's actually a p53 target gene. And then it dephosphorylates the whole pathway. So it sort of resets the pathway to baseline after the DNA damage response pathway is done. The mutations in ppm1d are always frame shifts in the last exon that remove a degron sequence. And so you retain full enzymatic activity. It's an active phosphatase that's massively overexpressed when mutated. And so it's constantly dephosphorylating this pathway, constantly keeping the DNA damage response pathway quiescent, and that actually helps cells survive, have a relative survival advantage when faced with DNA damaging therapy like chemotherapy or radiation. Sort of first part of this talk is that CHIP is extremely common. It's very age associated. It's associated with elevated mortality, which is related to increased risk of blood cancers, as well as therapy-related malignancies, increased cardiovascular disease. We've now started extending into other diseases, one of which is thrombosis, which specifically in JAK2 mutated cases is elevated. I didn't tell you the JAK2 story, but in that case, the mutant neutrophils lead to a process called netosis that leads to elevated risk of thrombosis. And TET2 mutations have an increased atherosclerosis in mouse models. We've now done this with DNMT3A mutant mice, and it looks like others are quite similar. And finally, the field is really extending a lot now into solid organs, and it looks like in many different tissues, this same process is happening where pre-malignant clonal expansions are extremely common. I think far more common than we might have guessed just a few years ago. So this is a really cool study from Peter Campbell where he took samples of skin that were removed for cosmetic reasons. It's called a blepharoplasty. It's a little sliver under the eyelid that is sometimes removed in older individuals cosmetically, and took hundreds of tiny, tiny little biopsies out of that bit of skin and sequenced quite deeply. And they found tons of somatic mutations, and not random ones. They're the specific somatic mutations that you see in skin cancer, not melanoma, but non-melanoma skin cancer. And if you put those mutations together in a cartoon format like this, you see almost a lawn of clonally expanded mutant skin cells with these driver mutations. Similar studies have recently been published by a couple groups in the esophagus, but I think we would predict that many of our solid organs, that particularly with stem cell function, will have these pre-malignant lesions that become extremely prevalent over the course of our lifetime. So, and you can track the types of mutagenesis that's happening here. It's clearly UV-mediated mutagenesis. In other organs, it's different, but the initiation of cancer is clearly not rate limiting. We have initiating mutations all over our bodies, for better or worse, and cancer only develops once you get those subsequent mutations in the same exact cell, leading to invasion and further expansion. These clones, unlike the blood system, can't get very big because they're spatially constrained. They don't have enough of an oncogenic mutation to actually lead to invasion, but they're all there. So with that, I'm actually going to switch topics a fair bit to another area that we've enjoyed working on recently of looking at thalidomide analogs and mechanisms of protein degradation. So you probably all have heard of the story of thalidomide. It was a drug that was developed In the 1950s, it was found to have anti-emetic properties, decreased nausea. It was found to have hypnotic properties, made people sleepy. And it was tested in rodent and models, in mice and rats. It actually had no toxicity, so it was thought this is a fantastic drug, wonder drug that has these valuable properties but no toxicity, so could be used in one of the most vulnerable populations and pregnant women who are suffering from morning sickness. It was used quite broadly in Europe, in Australia and Canada, and around the world. Thought to be so safe that it actually was over the counter in many countries, and took quite a while to recognize that women who had been taking this during pregnancy had an elevated risk of having babies born with phocomelia, these limb bud defects. In the United States, in contrast, the drug arrived on the desk of a very junior pharmacologist named Francis Kelsey, who said that, I don't see any data saying it's completely safe in pregnant women, and I'm not going to approve it. In those days, the pharmaceutical company could just call their bosses and say, well, that's ridiculous. It's approved everywhere else in the world. Every week you delay this, it's costing us money. But she really single-handedly held firm long enough till these reports of phocomelia started coming out. So the biggest prize at the FDA every year is the Francis Kelsey Prize, based on thalidomide, and the modern FDA that all drug companies need to navigate to get their cancer drugs and other drugs approved is really based on the checks and balances that were put in place because of the thalidomide experience. So it was obviously withdrawn from the market, but research continued. It turned out to be a powerful TNF-alpha inhibitor. It actually works in a rare syndrome called erythema nodosum related to leprosy. But it was eventually tried in clinical trials because it was thought to have anti-angiogenic properties, and it had striking activity in multiple myeloma, really not predicting why that disease might be a good one for the drug, but it worked. It got FDA approval. Minor derivatives called lenalidomide and pomalidomide had much greater activity against multiple myeloma. And amazingly, from one of the worst stories in the history of drug development, lenalidomide is now the top-selling cancer drug in the world, one of the top 3 drugs in the world in terms of sales. It has amazing efficacy in a number of B-cell malignancies. And it turns out that lenalidomide has particular activity in myelodysplastic syndrome with deletions of chromosome 5q, which, as Hannes mentioned, happened to be the very specific subtype of disease that I'd studied as a postdoc. So we were very eager to understand why that particular subtype has a dramatic response and to understand the biological basis. So didn't know what it binds to. A group in Japan, Hiroshi Honda's group, took thalidomide, immobilized it to a bead, pulled it down, did mass spec. We did the same thing with lenalidomide. Both of us pulled down an E3 ubiquitin ligase complex. There's a protein called CRBN or cereblon, which is the substrate receptor for the E3 ligase. As a reminder, as you probably know, E3 ligases are the enzyme complexes that target particular proteins for destruction. The substrate receptor, in this case CRBN, is the part of the complex that recognizes a particular protein that is going to be destined for destruction. So we have a compound that binds an E3 ligase. We didn't know what it did. We did mass spec and expecting to see some proteins have increased levels because most drugs work as inhibitors of enzymes. And if you inhibit an E3 ligase that leads to protein destruction, some proteins should accumulate. But we couldn't find any proteins that would recurrently accumulate in our mass spec data. But we did see proteins that had decreased expression, which was puzzling. But they validated, and the particular proteins were transcription factors. Icarus and Aiolos, IKZF1 and 3, are lymphoid transcription factors, and we were puzzled why they went down. But in Western blots, these proteins have decreased expression within hours of treatment with fairly low doses of drug, 100 nanomolar. And though this was, again, the opposite of what we would have expected. By IP mass spec, if we pulled down cereblon, this substrate adapter, in the presence of the drug, we could see the whole complex that we knew about, but also we could see these proteins, IKZF1 and 3, that we could pull down in the presence of the drug, indicating that they are bound to the ubiquitin ligase. The drug is present. So through a lot of biochemistry, which I'll spare you right now, we found that lenalidomide binds to CRBN When it's bound, there's increased affinity for particular protein substrates, including IKZF1 and 3, that leads to their polyubiquitination and proteasomal degradation. And IKZF1 and 3 are master regulators of lymphoid development. Myeloma cells actually are addicted to their ongoing expression, and their degradation leads to killing of myeloma cells. That didn't explain why it worked in del5q MDS, which is why we got into this field. But we did more proteomics and found another substrate called casein kinase 1 alpha. That's actually one of only 40 genes that's recurrently deleted in this syndrome. And these deletions are always heterozygous, never homozygous deletions or mutations. And so casein kinase 1 alpha is expressed at haploinsufficient levels, right at 50%, both at the RNA and the protein level. And full degradation in these cells kills the cells. So that led to this hypothesis that haploinsufficiency, 50% levels, would sensitize cells to further degradation, because if you get below a certain level, the cells would die. And that's actually a concept that had been out there for a while. And in the Broad, it was called cyclops genes. They were genes where haploinsufficiency sensitizes to further inhibition, but hadn't been shown in an actual drug that works in cancer. So we validated that this is a bona fide substrate of the ubiquitin ligase. This is an in vitro ubiquitination reaction, and we only see ubiquitination of casein kinase 1 alpha when the drug is present and the E1 and the E2 enzymes are present. So we actually really wanted to nail this. We made a mouse model of casein kinase 1 alpha, conditional knockout, characterized it, set up the experiment to show that heterozygous loss of casein kinase 1 alpha would sensitize cells to lenalidomide. Big experiment, lots of mice, and absolutely no effect. No toxicity of the drug, no efficacy, very sad postdocs. So then we did an experiment that somehow hadn't occurred to us before, which was a cell culture experiment in mouse cells. We'd always done our studies in human cells, but we looked in mouse cells, and these are mouse leukemia cells, BAF3 cells, and we treated with lenalidomide, and casein kinase 1 alpha levels didn't change at all. So we weren't getting any activity in the mouse cells. Then we did the thing you're supposed to do right at the beginning of experiments, which is to read the literature. And we couldn't find papers that describe the activity of these drugs working in cancer models in mice, and given they're really effective cancer drugs, that seemed a little unlikely. And then if we went back into the historical literature, which is harder to find, we noted that the drugs, that thalidomide had been tested in rodent models without evidence of toxicity, raising the question that maybe these drugs just don't work in mice, but our mechanism might be still correct. So now we understand the mechanism. We know the direct target of the drugs. So we could treat normal BAF3 cells with lenalidomide, nothing changes. Overexpress the murine cDNA, nothing changes. Overexpress the human cDNA for CRBN, the direct target of the drug, in mouse cells, and now the drug works. Casein kinase 1 alpha levels go down with lenalidomide treatment. We systematically altered all the amino acids between human and mouse and found a single amino acid change, I391V, in the mouse cDNA, changed the human residue, expressed that in a mouse cell, and it works. So a single amino acid potentially explaining a species difference in drug response. That seemed kind of amazing, that a single amino acid, particularly isoleucine to valine, a single methyl group difference, could make that difference. In crystal structures, we actually knew that the mouse CRBN bound the drug, which is actually why CRBN-based Protax works in a mouse cell. But this creates this steric bump right here, which is right where the substrates bind. And so the substrates are unable to bind the mouse CRBN in the presence of the drug and make all the direct contacts with the drug and with cereblon that mediates selective degradation of these substrates. So we can now understand why the drugs don't work in mice. We made a mouse that knocks in this single amino acid into its germline. It's healthy, doesn't have a phenotype. But if we do proteomics on their blood cells, these are c-KIT positive hematopoietic stem and progenitor cells. This is in wild-type mice. We would see proteins with decreased expression here. Nothing is significantly altered in wild-type mice treated with lenalidomide. If you treat the knock-in with lenalidomide, our favorite proteins, casein kinase 1 alpha and Ikaros, are selectively degraded. If we treat with pomalidomide, interestingly, pomalidomide does not degrade casein kinase 1 alpha, but it's a great degrader of IKZF1. These are not lymphoid cells. They don't express IKZF3 at all. And another protein, ZFP91, which we knew to be degraded by pomalidomide. So we now have a mouse model that responds appropriately to these drugs. We can do a variety of experiments. We validated that heterozygous loss of casein kinase 1 alpha sensitizes cells to lenalidomide. We could also look at pregnancy in mice. Turns out that this amino acid position lines up perfectly with the species teratogenicity. Primate models have teratogenicity to thalidomide, but one other organism, rabbits, have teratogenicity to thalidomide, and they have the human residue at this position. One primate species, the bush baby, does not have teratogenicity, and it has the murine residue at this position. So we took our knock-in mice, treated pregnant mice with lenalidomide or thalidomide, and the main effect that we saw in this initial study was loss of pregnancy. And some of the literature would suggest that was actually the major effect of thalidomide during pregnancy, but since it was generally taken during the first trimester with initial morning sickness and loss of pregnancy is so common in the first trimester, it was never documented, and it was an over-the-counter drug. Nobody paid attention. But it was probably the dominant effect of thalidomide. We now think through work from Eric Fisher's lab that we have the mechanism for the limb bud defects is a protein called SAL4, another transcription factor that has already been demonstrated to be important for limb bud defects and is a target of thalidomide and lenalidomide. So can we expand this concept of drug-induced degradation to other proteins? Our first step was to find the exact sequence that's necessary for drug-dependent degradation. And we did this starting with the minimal degron sequence we had, which is about 60 amino acids. We cloned it in this vector that we're super fond of using, where we clone this sequence, this degron sequence, in frame with GFP, followed by an IRES M-cherry, so that when we treat with a drug, GFP will go down because the degron confers instability to GFP. The M-cherry stays the same. So we did a saturation mutagenesis screen where we changed every amino acid in the 60 amino acids to all 20 possibilities and screened them with all the different drugs. And you can see that the same amino acids were important for response to each of the drugs. And the amino acids that were important, shown with these down blips, were within a single C2H2 zinc finger. And the only amino acids important on either side were structural parts of the adjacent zinc fingers, cysteines or histidines. And within this single zinc finger, each of the cysteines and histidines that define the zinc finger were essential, but there were other amino acids that were important as well. So we validated this in the same vector. And here is the full-length IKZF3 or iolose. And you can see the full length leads to beautiful drug-dependent degradation, almost complete degradation in response to lenalidomide and pomalidomide. If we knock out cereblon, the direct target of the drug, no activity of the drug. If we knock out the single zinc finger, no activity of the drug. If we clone just this single zinc finger by itself, we get beautiful drug-dependent degradation. If we mutate that zinc finger or knock out cereblon, there's no effect. So we have a single zinc finger that's necessary and sufficient for degradation by thalidomide analogs. With Nico Tomei's lab, we got a crystal structure of pomalidomide in complex with the single zinc finger from Icarus or Aiolos. It's actually the same exact sequence in Icarus and Aiolos, and cereblon, and could identify some of the contacts were made, one of which is this water-mediated hydrogen bond with this glutamine. If you mutate this amino acid, so this is with this amine that's present in lenalidomide, but not thalidomide. And if you mutate this glutamine, you end up with equal efficacy, equal potency of lenalidomide and pomalidomide with thalidomide. So that's one of the critical differences that makes lenalidomide and pomalidomide so potent compared to thalidomide and led to their phenomenal pharmacologic efficacy. So we can target a single zinc finger with a drug leading to degradation. Zinc finger proteins are the largest class of putative transcription factors in the genome. A bunch of them are involved in human disease. Is this a broader concept or not? So we explored the entire C2H2 zinc finger proteome by synthesizing all zinc fingers in the proteome, about 6,500, cloning that into this same vector in frame with GFP and M-cherry, treating with various drugs and seeing, can we induce degradation of other zinc finger proteins? And it worked very nicely. So here's with pomalidomide. We had a collection of genes that are really powerfully zinc fingers that mediate drug-dependent degradation. It's actually the same ones for lenalidomide and pomalidomide. Thalidomide's always the weakest, but the same findings. Out of that 6,500, we rediscovered Icaros and Ilos, which is reassuring, but found a bunch of others that are also have decreased expression after treatment with the drug. Some of them had antibodies, and we could validate by Western blot that we could degrade ZFP91, ZNF692, 276. Unfortunately, essentially nothing is known about any of these new transcription factors, so we have the complete opposite drug discovery problem where we can hit a target, degrade it in a transcription factor at that, degrade it very potently, but we have no idea what it does. Some of them might be responsible for some of the activities of the drugs that we don't understand, like the antiemetic effect. There's neuropathy from these drugs. There's a bunch of other properties, but we have to start from the beginning and doing ChIP-seq and understanding what these transcription factors do and would it be valuable in any human disease to degrade them. We've developed mass spec approaches to do just targeted mass spec for a small number of substrates or proteins. So we can now do dose responses, multiple cell lines, multiple drugs in a way that we can't do for proteome-wide studies. And you can start to see that these drugs have really distinct properties. CC885 is a new drug reported by Celgene that is degraded, induces degradation of these proteins GSPT1 and GSPT2, but none of the other drugs touch that substrate. Casein kinase is hit by lenalidomide and CC885, but not touched by pomalidomide or avadomide, whereas ZFP91 is very nicely degraded by avadomide and pomalidomide, but isn't touched by lenalidomide or CC885. So the particular activity of any individual drug will be the integration of all of the particular proteins that they degrade. The toxicities of the drugs will be dependent on all of those different proteins that are degraded. And just like kinase inhibitors have slightly different off-target effects for each particular molecule, these degraders are going to have slightly different effects based on the full spectrum of proteins that are degraded. Finally, just wanted to note that I described zinc finger proteins that are degraded, but I also mentioned two non-zinc finger proteins that are degraded, casein kinase 1 alpha and GSPT1. You might think that there are multiple modes of drug-dependent degradation, but it turns out that that's not true. They're all the same interaction motif. So in blue is the structure of IKZF1, zinc finger 2, the critical zinc finger, and in purple is the casein kinase 1 alpha, and the same is true for GSPT1. They both form a beta-beta loop with a critical glycine at the tip that mediates the interaction. And so the particular binding of the protein to the drug and to cereblon is actually the same shape and the same mode of interaction for all of those substrates that we that we know about. It's easier to find zinc fingers and interact in silico. Very hard to predict the beta-beta loops without a crystal structure, but we can identify these proteins functionally. You can do a lot of neat synthetic biology with this, including CAR T cells that you can turn on and off. We built both on and off switches based on this, but here's an off switch where we put the degron sequence onto the CAR T cell. And if you add the drug, you can completely degrade the CAR T cell. So if this is the expression of the CAR T cell, with the degron tag, you add lenalidomide and you completely remove it from the cell surface. So it looks like untransduced cells, but a CAR construct without the degron doesn't change. And that will alter the behavior of these T cells. You will block them from being activated in response to a target protein. We even made a super degron that has 10-fold greater sensitivity to the drug. So we now have FDA-approved drugs that are well tolerated that could act as on and off switches for various synthetic biology experiments, including CAR T cells, which can have toxicity when they become overactive. So the summary of this part of the talk is that thalidomide derivatives act as a molecular glue linking a protein substrate to an E3 ubiquitin ligase, and that degradation of different substrates leads to clinical efficacy. The thalidomide derivatives can target a particular zinc finger degron, degradation of Ikaros and Aiolos lead to efficacy in multiple myeloma. Degradation of casein kinase 1 alpha leads to efficacy in del(5q) MDS. And a single non-conserved amino acid between human and mouse can account for differences in drug activity in those species. And this is extremely collaborative work. Sid Jaiswal led the work on clonal hematopoiesis. Chip and Jan Kroenke led the initial studies on thalidomide analogs, but now have help from a wide range of people in the lab.